Good afternoon, thank you so much for being here. Today we will speak about the BIOforIU infrastructure, that is actually a multidisciplinary infrastructure to study and valorize marine and terrestrial biodiversity, everything in the framework of the Innovation Union.
BIOforIU is a center for experimental research on biodiversity and ecosystem, and has been built as a physical infrastructure collecting different type of data on biodiversity and ecosystem functioning, going from biochemical data, physiological data, to individual behavior and energetical data, and data on population, community and ecosystem. As a strength point of the infrastructure, it is composed by a number of micro and mesocosms available for the research on aquatic ecosystem, where different biotic and abiotic characteristics can be controlled and also controlled by remote. So scientists from their own laboratory, wherever in the world, can control the condition in which the experiments are run here in Lecce.
The aquatic system for experimental simulation provides facility for experimental works, for example, the implication of body size in the community of invertebrates, the bioecological response to ecotoxicological stress, and so on.
Part of the mesocosm is also conceived in order to work on climate change, including two ecotrons, a number of climate hazard rooms, and all the aquatic system where temperature can be controlled in very precise detail.
Amongst the various systems for housing model aquatic organisms, there is the zebrafish facility. It consists of 4 cabinets with 50 aquaria, each with a capacity of 3.5 or 8 liters. More than 2,000 fish can be housed in the system.
The facility is mainly focused on exploring biodiversity and morphological traits of phytoplankton organisms by confocal microscopy. The idea is to explore this method as a new frontier for both taxonomy and ecology studies.
We also have facilities for the study of bioenergetics, of chemical composition of individual bodies of cell, or also chemical composition of the aquatic medium in the environment.
The software itself is designed and allows to transform and translate the videos into data. Information regarding specific behaviors that may be related to taxonomy or toxicological effects or whatever else can be translated into data.
When the viral particles are very diluted in the environment, it is difficult to isolate them. For this reason, viruses are isolated from water samples by a tangential flow ultrafiltration system that allows to concentrate viruses and makes them available for biomolecular investigations.
Dangerous microorganisms are manipulated in Class 3 biosafety cabinet to avoid the possibility to spread of microorganisms in the external environment.
We perform both phenotypic and genotypic characterization of the microbial communities from various environmental sources, such as marine organisms, seawater, and sediments.
laboratory is really advanced. Here we can use the anaerobic chamber to cultivate, isolate and identify aerobic bacteria. One of the main aims of this laboratory is to not just enlarge our knowledge in microbial diversity, but to improve our biological application in the field like drug discovery and bioremediation of polluted environments.
Most of the facility has been concentrated in open space since research that can be done here are really interdisciplinary, involving many different fields of biology and ecology. And it's interesting that scientists and researchers can work together and can discuss together on topics at the multidisciplinary level. The infrastructure has been conceived as open to transnational access, and so is available to researchers, of course, in the University of Lecce and the partnership of BIOforIU, but also from all over Italy, Europe, and all over the scientific community.